So we'll now listen to the last 15 or 16 minutes, finishing up the last lecture. If you remember, we were talking about trying to determine when gene expression had period 2, and we decided that when this thing is very small, then it's periodic. And the reason for that is because we start counting off at I equals to 1 instead of I equals to 0. I equals to 0 was the first minimum and maximum that you would have if you have period 2. And then I equals to 1 would be the next one if you had more than one min and maximum. If you've got questions at any time, let me know. After we finish this lecture, we will talk about DNA topology, which will be covered in the December meeting. So, and if you've got questions, let me know at any time. If you notice any typos, I did, so I will type some down below when I notice some minor typos. Hopefully only minor.
Have a nice stability theorem. So remember, in terms of we would like that if two functions are close together, remember this is the soup over all points in your domain of f of x minus g of x, absolute value. So if f of x is close to g of x, then we would like that this thing to be close. So we would like phi q of f minus phi q of g to also be small. After all, our data is likely noisy, so if I perturb f slightly and create a function g, then I would like, this value to be close to this value. So if this is small, implying periodic 2, I'd like this to be small. If this is large, I would like this to be large, if we just perturb it a little bit so that this soup is large. Sorry, so that this soup is small. Well, we have a stability theorem for this total persistence. This is just some constant for a fixed q. It is some constant, so if this is very small, then some constant times a small number is still small, and so we do get that even if we change our function a little bit, we'll still get the same result. I don't want something that if I change my function slightly, that all of a sudden it becomes that it was periodic, but now after the slight change it's not periodic, or vice versa. And so these stability theorems are quite important. Otherwise, the results would not be valid. If you can do a slight perturbation, add a little bit of noise, and now come up with a different result, that would be bad. But the stability theorem says that if we perturb it slightly, then our function values that indicate the periodicity will not have changed much. Any questions up through there? Okay. So now let's take a look at this for q equal to 0, 1, 2, 3, and 4. What this is doing is up here is there are 27 genes that have been confirmed to be periodic. If we now take a look at the one that looks most like it's period 2, one that kind of looks period 2, so basically what we're doing is we're looking at increasing phi q. So we're looking at phi of q sort of increasing here. The larger phi q is, the less periodic we believe our function is. The smaller it is, the more periodic we expect it to be. Over here is the percent of the 27 genes that are found up into this list. So if I take a look at, between here, we find one of those genes when q is 0. If we are in this region, we might be finding 2 of the genes. Versus if we take q equals to 1, we start finding a whole lot more of the genes. 2, 3, and 4, there's not much difference between them. So the two, we start finding most of the genes. So if this is, so up to here, it does go up to 90%. So we found up to half the genes if we take a look at the top ranked genes right here. And I forget the cutoff for this one. I think this one might be the top 150 genes. So this is a ranking of the top 150 genes. So it is increasing phi of q, but it's the top 100 listed by gene. So the first gene that is most likely periodic, the second gene that's also very likely periodic, the third gene, etc. Actually, it's put 150 hatch marks on here, and it'll be quite close. So this is a little bit too close. So we might have had to have a few more genes right here. So maybe of the first, 5 genes, one of them turned out to be among this 27. The 2, 3, and 4 is basically what we're interested. This is, almost pretty much random. I go up to 150 genes, and maybe I find, some half of the 27. That's not very good. But the main reason for the 2, 3, and 4, we're interested in particular the 2. So Q equals to 2 is what we'll be interested in, simply because in terms of our stability theorem, it's stable as long as Q is greater than or equal to 2. If Q equals to 1 or Q equals to 0, then I do a slight perturbation. Then things will, perturb a lot. So for Q equals to 0, for example, I might have my, my DI minus BI to the 0 will either equal to 1 if DI does not equal to BI, and it'll be 0 if DI equals to BI, or if I don't have any, I don't have DI equals to BI, I might not have a sum whatsoever. So if I don't have any maximum or minimums, then if my n is equal to 0 and I have this nice periodic function, then my CQ of f equals to 0. And otherwise, I'm adding up 1 for each time I have a minimum and a maximum. I do a slight perturbation. I've now changed my function. So for q equals to 0, it's easy to see that it is not at all stable. Two functions that are close to each other, I'm counting the number of maximums I have or the number of minimums with this function when q is 0. Q equals to 1 is also not stable. You can find functions that are close to each other, but for where this is not small. So we really need to look at q greater than or equal to 2. And well, with q greater than or equal to 2, 3, or 4, there isn't that much difference in terms of finding things. So they focus mostly on q equals to 2, but they could have just as easily done q equals to 3 or 4. Questions up through here. Now let's compare the 5 different methods. So these are the 5 different methods. We're again looking the percent of benchmark genes identified. And in this case, the benchmark genes were 7 for this first graph. In the second graph, it's the same 27 we looked on the previous slide. The 27 were what was found by this L method. So this L method found 27 genes, which were then confirmed biologically. And these 27 genes, there's what's used to find here. And so thus, L found them all. So we could have done L, but L would have been, 100%, because that was the method that we're benchmarking against. We're benchmarking against method L. The 7 were known previously. They're clear. Everybody knew about the 7. And now we want to compare the old method with the 4 new methods. Well, you might think P stands for persistence, but we're interested in stable persistence. And so it's our S, the gray curve, that we're looking at. And if you look at the gray curve, if we rank them to the ones that we believe are mostly periodic, to the ones that we don't think are periodic, if we take a look at the top 300 genes, remember we had 7,549 genes. If we take a look at the top 300 genes, what percent of the 7 are in here? Well, for the gray curve is this one right here. So this is the gray curve. Maybe I should have actually used a nice gray pen for my gray curve. So my gray curve is this one right here. And you can see that for the 7 genes, it's basically listed within the top 100. So somewhere within the top 100, we actually find all 7 genes. Within the top, up to about here, within maybe the top 40, we found, say, maybe around 85% of the genes using the gray method. So that means that if we wanted to find these 7 genes using the stable persistence method, then we would have, if we wanted to find 85% of them, 6 out of 7, then we would want to do biology experiments on, say, the top 40 genes ranked by this method. To find all 7, we have to go a little bit further, but it does give you a ranked order to say which ones are most likely periodic. Oh, these are, so we'll do the experiments on these first to confirm. These over here, after 300, we don't want to check those. So depending upon how patient the biologists are, maybe they'll do 100 experiments, maybe they'll only do 40, maybe they'll only want to do 10, who knows. But it gives you a ranked order as to which ones you should check that would be most likely periodic and most likely involved in the somite development. If we then take a look at the 27 genes, well, with the 27 genes, we now get all the way here with going up to 90%. So in the paper they said 90%. I'm not guessing that. Everything else I'm guessing, but if I write it down, it's because it was in the paper. So they found 90% within the top 300 genes. And they got close to that earlier on. And so that's, the gray method we can see here still worked quite well in identifying these genes that were previously identified. Questions on this slide? On the last slide, well, these methods do find additional genes. So we do find more potential genes that might be periodic with the same 2-hour period. Let's take a look at the graph on the right first. So they ranked the top 300 genes. So we ranked the top 300 genes using the L method. Remember, it's the S method that we like. That is our stable persistence. And so if the biologists were willing to carry out 300 experiments, they can choose to carry out the 300 experiments found using stable persistence, or the 3 methods using the L method, which is sort of a modified Fourier analysis method, or one of these other methods. If you take the union of these, they do have things in common. So, for example, if I take a look at S and L, they have 135 in common. So of their top 300, they have 135 in common. But if I wanted to take a look at all of them, I'd be taking a look at 884 genes. So 884 genes were found using one of these 5 methods. That's a bit much to check out. But you can take a look at the intersection of the methods if you like, but they might have different, the stable persistence didn't really find this one. So maybe you don't want to throw some of these things out. If you look at what's in common with all 5, there's 21 genes that are common with all 5. So those should definitely be checked. What else should be checked? Maybe you want to do the top 50 and all of them. They didn't give the graph for that, so I don't know what the intersection is for that. We can take a look to see what is common among, I think this is gray, yellow, and orange. So gray, yellow, and orange. So this would be 58 are in the intersection of the L, P, and S method. And then you can take a look at what's in the intersection of 4 of them, etc. So that's what this thing tells us, is what is common to the 5 methods, or what is the union of the 5 methods. On the left side over here, what we have is the genes for the stable persistence. That is the top half. So all of these genes, if you add up the numbers, 0 plus 15 plus 20, all these numbers on the top half, you will get 300. If I take a look at the L method, so in terms of the L method, I need a yellow color. The yellow color is this, and so that would be this thing here. So if you want to see what's in the intersection between yellow and gray, that would be this right here. So if we add up these numbers right here, that should equal to the 135, the intersection of S and L. And similarly, we can, go with a purple color and take a look at what's over in here and take a look at the various different intersections. So that's another way using Venn diagrams to illustrate. So that's basically one method for using persistent homology. There are many other methods. You can see that no method is likely perfect, but it does give biologists an idea as to which genes they should test that might be associated with somite development. Any additional questions? Thank you.
No questions on that one then. So then let's go ahead and share this screen. So for the rest of the class, we will discuss DNA topology. The December workshop will talk about DNA, RNA, proteins, lots of biological applications. At least 2 of the speakers, most likely more, but right now I'm trying to confirm who's speaking about what, and not everybody has decided which topic they're going to talk about yet. But at least 2 have confirmed here, and there are likely others that will talk about DNA topology. So today we will brush up on our DNA topology. So you know that DNA has this nice helical structure, and that it's got these base pairs that, get this double helix together. Well, you can have, so there'd be lots and lots of base pairs, and in reality this is supposed to be twisting around, but just drawing it straight for simplicity. This right here is linear DNA, since the ends are not joined together. In the biology world, if there is linear DNA where the ends are not protected, then the ends will be eaten. So there are proteins that will degrade DNA. If you've got foreign DNA in your body, or if there's some kind of, your DNA gets split off through some sort of error in something, you want to get rid of that DNA, and so anything with unprotected ends will get eaten. There are DNA viruses that will start off linear. They will then be ejected into the cell, but they have what's called sticky ends. So this end right here could base pair with this. So assuming this is long enough, so if the DNA is long enough, these sticky ends will find each other and they will bind together, creating circular DNA. In your own body, your DNA is linear, But again, linear DNA will get eaten unless the ends are protected. So usually you have some very interesting structures. I'm trying to do a G quartet right here that will protect, along with a lot of proteins, that will protect your linear DNA. And then is also frequently tacked down to a scaffold and kept very nicely organized. And when you need the DNA, so normally the DNA has lots and lots of proteins associated to it, very, very compactified, but when you need the DNA, some will come out, giving you a chance to replicate or transcribe the DNA. So when you've got the ends fixed, you'll have similar types of problems that you have for circular DNA and also for long DNA. So on this side, we've got nicked DNA because the phosphate backbone here is broken. So if I take a look at this thing right here, it's not sealed. So the phosphate backbone between the GC and the CG, it's broken, and so it's nicked. That means it can have whatever twist it likes, versus down here we've got closed circular DNA, which will actually behave very differently than nicked DNA. Well, one thing to keep in mind about DNA is that it does have a chemical direction. So it's 5' to 3'. The 3' is connected to the 5' in this direction. It's the opposite polarity here. So the DNA is anti-parallel. To topologists actually find that sad because you can't create any Mobius bands. So there's no Mobius bands because if we had, when we tried to connect things up, then in one direction, if this is where you were trying to connect it up, they'd be going in the wrong direction. And so no Mobius bands in our DNA. So we'll only get nice annuli, but in reality the annuli will be twisting about each other if we think about our DNA as, so if I go back to this picture right here and think about this thing as forming the boundary of an annulus. So no Mobius bands. By the way, whenever I say that we never have something biologically, I never mean never in biology. They actually have constructed a Mobius band using DNA, but as far as it's known, it's not naturally occurring and it's very difficult to create. It's not the natural structure. So the natural structure will be this sort of annulus type thing formed, although in reality your DNA will be twisting around each other, just drawn plain for simplifications. Well, because your DNA does twist, we have this twisting, so we drew our two strands like this, but in reality we can have, let's see, DNA is right-handed, so this should be our basic twisting. So it's like a right-handed screw, so you can kind of fit your right hand in here. Your fingers would sort of run parallel to here as you're trying to, go around with your right hand if you put your then thumb over on that side. Yeah, that's a nice picture of a hand. So your DNA does have nice right-handed twists, normally speaking. It has a preferred twist that it likes to have. So it would not like to have no twist. It would not like to have, here we have a full 360-degree twist. You can basically figure out the twist by just thinking about if I take, this arrow followed by this arrow. If I look down vertically, they will be changing in terms, so I'll have a d theta, and then you can basically just integrate your d theta. So there's nice math formulas that you can get to get the formal twist of your DNA. One thing to note is that the twist of your DNA, when we calculate it, it is generally about the backbone. So it'll be generally about the backbone. So we'll do, say, the twist of this one If I were to take the two strands, you actually mathematically get a different number when you integrate this. So instead, we do, the center line. So if I take one of the phosphate backbones, so if I take the twist of one of the phosphate backbones across the center line, and that's the twist that we're looking at, not the twist of the two backbones about each other. So mathematically, there actually is a difference, and so we're doing it about this center line. But in either case, you can think about, I've got one 360-degree twist here, and so that would be one full twist. Here we have a half twist, and here we have a full twist over here, and here we've got zero twist. Any questions about twist? Well, DNA, so here's some pictures of DNA where we can see it looks like it's supercoiling on itself, and here it's not. In both cases, this DNA is actually has twist in it. It may not show twist, but it does. Your DNA, remember, is this double helix, but it's been coated with a protein to make it thicker so we can actually see this. Oops, I just drew Z-DNA, left-handed DNA. Sorry about that. This one's the right-handed twisting, so we'll get rid of that. So we should actually be, frequently when we draw a line, this line actually is supposed to represent double-stranded DNA. And in this case, there is a protein that has come on here and basically coated it. So you've got this protein that's gone ahead and coated this entire thing and makes it basically look like just one thing, but it is twisted up. Any questions about that? Well, DNA likes to have a particular twist. And if you close it up, when we go ahead and take a look at these things here, if I close it up so that it has its preferred twist, it would be happy. But if we close it up so that it has this very underwound twist, then it will supercoil upon itself. You can do your own experiments by taking a belt and twisting this belt, or just take any type of cord. So if I take any cord, take your nearest cord, start twisting it. If you start twisting it, it will start to supercoil upon itself. And so we do have the supercoiling upon itself. Any questions up through here? This supercoiling is considered to be writhe, and so in terms of this writhe, we can now count the number of times the cord twists about itself. I was told that if I only use the arrow keys, my mouse would not have any issues. Sorry, not my mouse. My laptop would not have any issues. I've tried to actually change the slide and was unable to do so. So now I will, it worked so nicely last time, add this to, okay, there's nothing on the screen, so I will go over and put it. Okay, I won't be able to actually see any of the comments because I'm switching to this computer for the slides. So if you do have questions, just speak up and let me know. So we will continue with this one. So here we can see that we do have our supercoiled DNA, and we just want to calculate its writhe. Well, to calculate the writhe, it will depend upon the signed crossing. You can either use your right hand, go underneath with your fingers in the direction of the arrow. So in other words, if I go back up here and put an arrow on this one right here, and then follow along with that arrow and put arrows on each of these, you can take, in this case, we would need to take our left hand and put it under in terms of the direction of the, actually right hand. So this is positively supercoiled. DNA is usually negatively supercoiled. But if you take your right hand, go underneath that crossing and have your arrow point in the same direction, that gives you a +1 crossing, and in the opposite direction, it gives you a -1 crossing. Alternatively, you can just rotate the over arrow to the under arrow. If you do that in the counterclockwise, or the counterclockwise direction, then it's a plus crossing. If you do it in the clockwise direction, then it's a minus crossing. And then to do the ride, you can just count all the self-crossings. Let's do a sort of tracing of this thing here. Well, if we assume that they are all the same type of crossing, I can rotate the over arrow to the under arrow. That's going counterclockwise, so this is plus. And so we have 1, 2, 3, 4, 5, +5 supercoils. So we have +5. The ride, in this case, would be +5. That means that the DNA is overwound. Generally speaking, the DNA likes to be underwound so you can pull it apart for replication and transcription. In this particular case, I'm going to think about this as my double helical DNA. So the blue and the red are my two different phosphate backbones, and I should be having about 10.5 base pairs for every turn. I actually now have more because this would be a full turn right here. What we now want to take a look at is what's called linking number. So linking number, we again do our two signs. And so if I put these arrows up here again, in this case I am rotating in the clockwise direction. So I've got a minus sign here. For our linking number, we add up the signs of all the crossings. Now between the two things, so we'll add up the signs of the crossings. So a crossing between our two links. Between the two links. And then we divide through by 2 because we want a full turn. So I've got 1, 2 crossings, so I can count just where blue crosses over red. I can get the linking number as -2 that way. I can look at where red crosses over blue, get the linking number of -2, or I can look at all 4 crossings, get -4, divide through by 2, and I get a linking number of -2. So that's our linking number. Generally speaking, though, we want to think of our DNA like this. And if I think about my DNA as this, then think about pulling this red over here. And now my DNA, so we get rid of that side here. Now you can think about the DNA here. You can imagine that since DNA has about 10.5 base pairs per term, the linking number will actually be quite high. Any questions up to here? And remember to ask verbally. Linking number can be 0. In this case, maybe instead of having each of these curves representing a single backbone, sometimes your DNA, sometimes these single curves will actually represent double helical DNA. So I might have one of these curves. So when you listen to the talks in December, you'll frequently have to tell from context whether or not a single line represents double helical DNA or whether it represents, 2 strands of your double helical DNA. And sometimes people actually work with single-stranded DNA as well. In this case, our linking number over here is -2 plus 2, so -2 plus 2 gives us a linking number of 0. Excuse me for So with our linking number, though, for DNA, we'll generally have pretty high linking numbers for our DNA. Now, you will notice one thing when you pull this thing over. I've got two strands that are actually parallel, so if I orient this and put my arrows in here, they're both in a parallel direction instead of an anti-parallel direction. We said that DNA was supposed to be anti-parallel, but in order to calculate the linking number, we just, for mathematical convenience, assume a parallel orientation. Questions on that? So for both these pictures, we have a rather high linking number. So it's a rather high linking number right here. But in this one, we also have ride. So we also have ride, where in this case, for the ride, we add up all the self-crossings. So we just do self-crossings here, only self-crossings. And for that, we take the center line. So we take the ride of the center line. So remember, we had our two backbones wrapped around each other. For the ride, we're interested in the center line. For the twist, we look at the twist of one strand about the center line. And for linking number, as long as you take any pair of these strands, you'll actually get the same linking number. Questions there? Well, there is a rather nice website with a that actually shows the linking number quite nicely. So let's open up that website. Okay. So if you go to this website, you can see how twist and ride can interconvert. So in this particular picture, we have a twist right here, a half twist here, a half twist here, for a full twist. Again, if you have a belt, you can take it off, put in a full twist, let it go, force it onto a flat table, and it will ride upon itself, converting to ride. So we will go through these things a bit quickly. So we're going to ignore the sound, which I had muted on the other computer. Maybe you can still hear me, but now we can see twist converting to ride. So again, notice how it's useful to think about this as a ribbon. And if I can draw the DNA strands here. Well, they won't be valid DNA, but I can draw them all so that none of them twist. So you can draw a bunch of vertical lines connecting the red curve and the blue curve together. And you can see thus that there's no twist here, versus here, if I draw a line connecting the red curve and the blue curve, it's going to twist by 180 degrees as it goes through here. I'm going to mute again so we can see the pictures while I fast forward. So now we can go back to here and take a look at this slide right here. Any questions or anything before I go back to that? Okay. So, so one can convert between twist and ride, but the linking number, you can see from our linking number thing, unless I break one of the strands, unless I break, cut this across, there's no way to unlink these. So the linking number is a topological invariant. So if I deform my thing without breaking anything, the linking number will remain constant. We did see, though, in the demo that my ride, we could untwist this thing a little bit, change the ride, and convert things into twist. And so your DNA, it's constantly moving. Twist gets changed to ride gets changed back to twist. But we do have a nice formula that link equals to twist plus ride. Now, I was a bit sloppy with my definition of ride. You'll notice, if you go back and look at that video sometime, you'll notice that twist and ride never went to 0. And that's because if it is crossing itself, if I've got something that's crossing itself once, when you actually do, this is the 3-dimensional ride. You're supposed to project this over all dimensions. And so in most dimensions, if I project this, if I take this 3-dimensional object and shine a flashlight in, a variety of different directions, in most directions I will see this crossing. But if I look basically along here and project there, well, I'll get something that, or let's project from the other side. So if I project from this side here, it's going to basically look like this with no crossings here. So the ride of this thing will actually be almost 1. Again, I can check the sign of that by rotating clockwise, which means that it's a minus or a, so clockwise. So it's either a plus or minus 1. I can't do the signs anymore. But so that on average, we'll see 1 crossing, but we won't always see 1 crossing. And so the absolute value of the ride will be slightly less than 1. Similar for twist, twist does not mean to be an integer. And so we may have some twist in here that will probably have a lot of twist, but it probably won't be exactly an integer in terms of how it's twisting. Link, however, has to be an integer. So this has to be a whole number. We can't have half a linking number. And that goes back to, if you take a look at our definition right here, I have to go back, and we're not getting any Mobius bands here. So when we connect back up here, we're going to get a linking number of a full minus 1, or of a full plus 3, or of a full however many crossings, 1, 2, 3, 4. So a linking number of 4 right here. And so that's an integer and the topological invariant, and the two can interchange between each other. any questions up through there. So now we can take a look at what this means in terms of replication. Well, this twisting, we have to pull, when you replicate or even transcribe, we have to pull these two strands. They have to come apart. So this is an example of replication where these two strands have to be pulled apart. Well, that has interesting consequences. We have to unwind these two things in order to replicate. So these two things have to be unwound. Then they have to be, rewound as you're going through your replication. You're unwinding up front, getting these pre-catenanes in the back. And so your final product will generally be interlinked DNA rings. So the thing isn't fast enough to remove, and getting to zero is hard. It isn't fast enough. So this is your newly replicated portion. But your newly replicated portion will have these pre-catenanes, and you can see that in here. That this actually is double-stranded DNA that has been newly replicated. This part has not been replicated yet. And it is linked. And when it comes to the end, you will actually have two DNAs, two double-stranded DNAs that are linked together, and thus you need a protein that will fix it. And so we'll talk more about the topoisomerase II protein, that will break. So now this strand here does represent my double-stranded DNA. And it will break one of them, allow the other strand to pass through. And so when we used to have the green crossing over the aqua, we now have the aqua crossing over the green, and that is the way the DNA can be unlinked. Topoisomerases are vital in replication, transcription, unknotting, unlinking, and supercoiling. They're the targets of many anti-cancer drugs as well as many antibacterial drugs. Fortunately, I'm not taking any antibacterial drugs at the moment for my cold, but if I did, I would look it up and find out if it is a toposide. So topoisomerases are very necessary for replication and transcription, and so if you want to hurt your bacterial cells or your cancer cells, especially your cancer cells which are rapidly replicating, then if you slow down the topoisomerase or the toposide, then that can kill your cancer cell, as well as hair cells and other things, that stomach cells that are also replicating, and thus you can also get these side effects from your antibacterial and anti-cancer drugs that are affecting the topoisomerases. So we will talk more about this next time. So sorry for the technical difficulties again, but hopefully we will have better luck next time. And I'll have more backups going too. Any questions? Well, see you on Monday.